H-bound networks for almost 2 years, not quite. And I think both of us came with similar thoughts and intentions and hopes. And me coming from Duke, obviously Duke having been a focus of big problems with reproducibility and transparency in terms of clinically applied genomics. Brian having been, luckily, Mayo not having that kind of scandal, but being interested in those same things. I think a lot of what inspired this pilot project is us talking about things, because when I came, Synapse was pretty embryonic at that point, but it was already a REST API. Basically, as probably most of the participants know, providing unique web links to pieces of data. And then really in our first year here, a lot of progress was made in developing a programmatic client for using that REST API within the R computing environment. So we tend to use R a lot here at Sage Networks. One, because it's an open source, higher level programming language, and two, because it's very good for doing the kinds of genomic analyses that we're interested in, because actually down the hall from us is Bioconductor, and they have a lot of great tools for genomic analysis. I know probably many of the people involved in NIF are working more on the cell biology side of things and use different languages, but that's sort of where we've started from. And basically what transpired is that I was thinking about a manuscript that I was working on, and how can I embed what we're doing with Synapse into that manuscript, or vice versa? Or should I even be thinking about whether a manuscript should be a standalone entity of itself? So that's what inspired us to start thinking about how can we knit all this together? How can we make every part of what you would call a manuscript, whatever we want to call it, a scientific communication, web accessible? And as Synapse matured, as our client that we use for using Synapse matured, we thought we had a moment of opportunity just to be very pragmatic and practical about figuring out how do we think about basically erasing that border between actually doing computational scientific work and actually making it communicable. Not as in the disease, but as easily communicated to our peers and public. So that's what inspired us. And so basically, what you're seeing here is a web page which represents our technical demonstration of how we might do that. So can people see my cursor? Oh, I guess Anita's muted, so she can't tell me. But anyway, hopefully you see my cursor. So this,
yes, I can.
in many ways, okay, looks similar to the front, page of a scientific journal article on the web, but it's a little bit different in that, yes, it has a title, it has an abstract, it has authors and things like that. But what's different here is, besides just the sort of textual description of a project, are basically those components available via Synapse of the paper. So this fancy looking graphic here is a provenance graph showing that we've taken a breast cancer data set, we've generated training and validation cohorts from that breast cancer data set, and we're building models. And basically, each one of these pieces of a paper, whether it's a piece of data, a piece of code, a piece of intermediate data, are linked in terms of their relationship to one another. And this is a nicer pictographic, illustration of that. Right now, we have the REST API for providing this provenance graph, and hopefully, within this year, we'll actually be able to programmatically generate this and have it look good But that's basically, right now, I hand-coded this particular provenance graph. The other components of a paper are the pieces of code, for instance. So we have on the right side column web links to pieces of code. So I can actually click on one of these web links and it'll show on Synapse, assuming Synapse comes on quickly. Oh, I guess we have to log in. We'll log in as Brian. So let's do that again. So we can see that piece of code living on Synapse. How come it's not? Or we can actually show it in RStudio. So one thing is, one great tool that we've been using is an open source software project called RStudio. So since we use RStudio is a really nice technology because it's an integrated development environment that's actually web accessible. So basically, you can run a virtual machine in the cloud, whether you're using Amazon, Google, whatever flavor of cloud computing you'd like, and you can actually host basically an integrated R development environment through a web browser through that. So what I'm going to do here is I'm going to click on RStudio, and it's actually what it's going to do is it's actually going to load that piece of data into RStudio. And while we wait for that, I'll go through. So basically, we're providing web links, all these things. And we're also showing how you can access that by having this button here is hand me your cloud computer, because when I'm talking about what erasing the border between doing your science and actually presenting it is really the ultimate way to do that is to be able to actually hand your machine to someone. So we know that with intense computational work, even if you describe everything that you do, it's often very difficult for people to reproduce exactly what you're talking about. So why not use cloud computing and virtualized machines to basically provide a way for someone to hand their computer to someone else? So going back to RStudio here, what it did was it actually loaded that piece of data into the RStudio integrated development environment. And this could be made basically if we were submitting this manuscript for review, a reviewer could actually start playing around with your analysis by actually just clicking on that link to RStudio or the hand me your cloud computer link. So what I also want to show is how we can knit together an entire analysis. So we're generating models, we're generating figures, we're generating intermediate data. So how do we knit all these things together? And what I'm going to do is I'm going to step through basically all the steps in generating this pilot or technical demo. So in this case, what we're doing is we're taking a breast cancer data set, and we're doing something relatively simple in terms of in the breast cancer biology world is we're just trying to develop a model of estrogen receptor status in a training set and then validate it in a validation set that's held out. And we're going to knit together this analysis, and what we're going to knit together with are the Synapse client that I mentioned to you. So if you can see my cursor, we're basically bringing in the Synapse client library and this other client which Brian wrote called the R GitHub client. So this allows us to move back and forth between Synapse and GitHub. We talked about sharing code. Well, one way to share code is we can actually have code web accessible in Synapse itself. But if you're developing something and you want to have that flexibility because it's still a work in progress, it's nice to keep your code in a version control repository that's web accessible like GitHub. So that's what we're doing here is then that we're bringing in that ability to knit those two things together. And what we're doing is we're actually bringing in the ClearScience web repository into the R environment. And what I can show is the actual code repository as an R object now in our integrated development environment. And in fact, we can even view it. And so right now, just using that command in RStudio, we're actually accessing in GitHub the actual code repository. So it's a nice way to move back and forth between working with code, even if it's in progress, and allowing other people to see what we're doing. So we're going to keep on stepping through. I'll cancel that because we're going to move to the next step. And what we're doing is we're actually sourcing specific pieces of code from that repository. And we're running them now. So what you can see in this window on the bottom right is it's actually loading the data set and actually randomly splitting it into training and validation sets. I can also show you the names of all the variables that have now been populated into the R working memory. And now I'm going to step through the next steps of this analysis, and basically now it's running the code. It's actually running a random forest model on the training set. So it's basically training on estrogen receptor negative versus estrogen receptor positive patients and developing a random forest model based on that. And then we can look at the, oops, we can look at the variables that have now been added to the R workspace, and we can actually look at the figures that were generated. So we're reproducing this analysis on the fly. Certainly, if I were a reviewer for PLOS Computational Biology, I actually was poking Phil a little bit one time because I said, well, I was asked to review a paper for PLOS Comp Bio, yet I got a PDF. And obviously, we want to move beyond the PDF in this case. So this is one way one might think about doing that. So stepping through the rest of the analysis, again, showing you the piece of code. So this is actually the code for validating the model. So we can move back and forth between GitHub and ClearScience and RStudio very easily. Again, again, bringing in the specific piece of code and running it, we can look at the variables that generates, which include figures. So now on the upper left here, you can see how the model looked in the validation data set. So it looks like it worked pretty well in this case, and I didn't spend much time tuning. You can also show alternative ways of visualizing that analysis. So in theory, what you can be doing is every figure that you generate in your paper, you should be able to generate programmatically by knitting through the analysis. So here's the receiver operating characteristic curve for this particular model, and that's sort of the full code demo of what we're doing with ClearScience. So click on components of this provenance graph and then actually populate that into the RStudio environment as well. So everything is clickable because there's no reason why every component of your paper shouldn't be clickable. Another thing I want to highlight is that a nice model that GitHub has promoted is forking a project. So, and software engineers actually do this all the time now. And in theory, well, let me explain to you what forking a project means. To fork a project is basically cloning that project and creating a link to that project on GitHub. So then if you're interested in something and you want to continue working on it, you think, well, I have an improvement I can make on this project. You can fork it and you can make your changes. And since it's a version control repository, you can actually make those changes without worrying about whether you're deleting someone else's work or overwriting it, et cetera, et cetera. And then what you can do is, well, I've got a significant improvement to this project, and this is certainly the way programmers at Facebook and Twitter work. And then you can actually file what's called a pull request. So whoever's in charge of the project, you could say, look, I've made some improvements. Will you please look at them and consider integrating them into the main developmental stream of what you're doing? And Brian and I both think of this as really what we should be doing scientifically, right? The scientific method is a constant history of forking projects. If someone developed a southern blot, gosh, now probably more than 50 years ago, then other people can elaborate on that and develop the northern blot or something like that. So science is a sequence of forking, and let's use that model that GitHub has used. So in fact, we can click on that. And in fact, since I'm on Brian's computer, I can actually fork this project to Brian's GitHub account. Um, and Brian certainly chime in with it if you have anything else to say. Another advantage of using a version control repository is, let's say you've gotten your project to a point where you say, okay, this is the part, this is the way my code and everything looks that I want to submit it to a journal. And you can actually tag it, so you can actually programmatically assign that milestone to that where you are in your development and say, okay, now this is my version 1 or my version 0.9-11, and this is what I'm submitting to Nature Genetics. And assuming they accept your paper and everything like that, you can say, okay, this is where my project stood at the time it was submitted and accepted by Nature Genetics. So people can always go back to that specific milestone, even if you might be actually moving forward with the work. That represents the kernel of some new analyses that you think might be useful either in the clinical world or scientific world. Now you can move on to version 2, but people can always get back to version 1. And again, that's taking advantage of the metaphor that version control repositories give you in terms of tagging things at specific points in time. And when someone forks a project, then they can develop an alternative branch of your analysis, and that can be tagged, and then we can always keep track of what other people are doing based on that original code base, and that's why we talk about version control trees. So we like using that metaphor. So this demo is in fact what we showed to Josh over at the R.P. Sloan Foundation, and indeed that's part of the reason is, that this pilot project was funded, was having this pilot project, this demonstration ready for him. Brian, are there other things you think I should highlight?
No, I mean, I think this really shows a vision of where we were hoping to head, and Synapse, as you can see in the upper right hand portion here, we talk about Synapse powered building blocks. Synapse itself is rapidly acquiring tools to be able to put all of this together in a way that is not a static page, but something that can in fact be developed by anybody utilizing the system. So
yeah, Brian and I have been using this as a battering ram to sort of well, I shouldn't use that military term, but we've been using it as a way to help the software engineers here, think about how the REST APIs. Basically, I talk about this provenance API. We actually have a working provenance API that just came online a month ago, and that uses the W3 spec for provenance. So it's using an established, well, I guess it may not even be quite at spec level, but it's the final recommendations of the W3C provenance group, working group. Obviously, we have REST APIs for accessing all the pieces of code and data, and then we're leveraging GitHub and RStudio to help sort of knit all these things together. And I guess where's the Adobe page? Yeah, the Brian. Yeah, one other way of presenting things. So obviously, presenting things the way that we've shown you is a way that obviously is more useful for someone who feels comfortable in a programming environment, but not everyone does. And in fact, we should think about engaging our entire audiences. So for me, someone who's been working in the translational genomics, and how do you make that relevant to patients and clinicians, a clinician is not going to want to step through your analysis. They want to be able to take it off the shelf and use it. So another thing we've been playing with is this. So here's a similar project to the ER pilot technical demonstration that I showed you. And in this case, we're using yet another piece of technology developed by the RStudio guys. These guys are good. And what we're doing is an analysis that I did just like that ER analysis with the provenance and everything, all the pieces of data backing it. What I've done is I've created a web application for kinase pathway in breast cancer. And what I can do is I can actually make it so that in real time you can actually take a patient's data, submit it to, again, in this case, a random forest model. And what it's doing is generating a activity score for the PI3 kinase pathway up here. And it's also plotting that patient on the far right here in relation to a bunch of patients developed from who are provided via the Cancer Genome Atlas project. So I'm actually plotting that patient in comparison to wild type patients, patients with known mutations in the PIK3CA locus, and other PI3 kinase related loci. So for a clinician, this would be interesting because then they can say, okay, if I have PI3 kinase pathway targeted agents, and I can actually plot my patient in relation to a whole population of patients, now I can actually play with that concept. I may not want to play with the ClearScience, stepping through all the code and all that mumbo jumbo, but I can basically take a model living in Synapse, accessed in Synapse via the REST API, and actually use it. So that's another component that we're now working on as well.
And just
oh, go ahead.
I was just gonna just to elaborate on that a little bit. A lot of what we're trying to do is providing readers or consumers of science a varying level of abstraction away from the actual science. So we completely understand that not everybody who comes into a scientific project is going to want to sit down at a command prompt and interact programmatically with all the work that was done. Some people may just want to read an abstract. Some people may just want to look at the figures. Some people may want to interact with the data with a web app like this, and some people may actually want to take the entire project, fork it, and go work on it themselves. So providing those different layers of abstraction really, we're trying to tailor this to as broad an audience as possible.
So this is many of these pieces are absolutely spectacular. You're welcome to. I don't have any specific questions from the audience, so I'm just going to start us off here. So what I was seeing is a bunch of your code and a bunch of these things, and the whole idea is fantastic. But what I got a little bit lost on is, okay, so if I have my data set, how do I know that my data set is going to fit any of these analyses that you had? What is the way that a clinician would be able to, add patient C or D?
Yeah.
How would you know? So that would be, one thing. And then I'll ask you some really hard questions later.
Well, that's a hard enough question. That's a constant question in terms of computational sciences. And obviously, at Sagebot Networks, we think about this all the time. How do you make disparate data sets fit together well? And honestly, you may think it's a cop out, but the only way I say we can actually move closer to that point where that becomes easier is by making things available, openly available. I think part of the reason why we have so much trouble doing that is people are so intent on holding that data close to their vest because it represents an academic advantage over their competitors. And so there's almost a disinterest in making sure that data sets can be locked together. And then all the circumstances with data sets, we all know, especially with genomic data, is that the process of generating it, the batch effects, the technician related effects are often latent in your data sets. So, and it's very difficult for people to explore them because they're not available. And by making your analysis flow available and transparent to people, then they can understand, okay, so they normalize their data this way. I see that in this case, they actually did identify that a site where a particular part of the data were generated is actually exerting a lot of batch effects in the data. And you can actually see that they compensated for it programmatically in a certain way. But I do admit that I'm copping out, and that I don't want. I figure at this stage, imposing standards is probably fraught with problems because we don't know what the best standards are, and it's only in a world where everything becomes very open and transparent can we judge for ourselves what ought to be the standard. So that may be a cop out answer, but and Brian has something to chime in.
Yeah, I'll jump in there as well. If we take a look at the PI3 kinase pathway activity prediction here, so this is based on, I'll go a little bit more technical than Eric did, and just talk about the shiny infrastructure that this is based on. So we have a drop down where you choose a patient, but one thing that they have provided as well is the ability to in fact upload your own data into this. So you can imagine having a web accessible app where you would say, as a clinician, push a cell file full of patients' genomic data. And as long as the model knows what to expect, as long as the model knows to expect this sort of file, it could handle all of the processing and spit out a prediction custom to whatever that file uploaded was. What Eric was alluding to surrounding batch effects and that sort of thing, those are really, really important research questions that need to be figured out for this sort of translation to really be able to make any headway.
I don't think that's a cop-out answer, but I think we've got, okay, so I will hold my questions because I have a question for Marianne here. All right, how do you envision this fitting into the current scholarly publishing landscape? Is this meant to go along with a traditional publication, replace it? Are you working to have ClearScience indexed in PubMed?
Yeah, so part of the pilot project, working with Josh and the Sloan Foundation, was also engaging publishers in the conversation. And so that's actually, in fact, how we became, started talking a lot with Phil Bourne. We've also been talking, basically providing an external advisory committee for what are the issues that we face publishers. So Miles Axton at Nature Genetics, Kelly Lamarco at Science Translational Medicine, the guys at eLife, Mark and Ian. And yeah, we have to think this through because, and we also have to think about it, how do we fit in the context of publishing? Our fantasy is one day, who knows, if you can show your analysis and make it web accessible, I mean, where does the publisher, fit into that? And certainly we just want to be flexible in terms of figuring that out because obviously you can see the editors that we've engaged include editors of open access publications as well as closed access publications. I think we would like it, at least initially, to be basically a way alongside traditional publications to present your work. And in fact, with the current breast cancer challenge that we finished running, and basically that was a challenge where a bunch of competitors from the public, all these are machine learning type people, were provided access to a large breast cancer data set and then developed models to validate in a newly generated data set. And that the winner of that challenge actually gets to publish a paper in Science Translational Medicine. And in fact, we're going to make a ClearScience version of their work and have it available as a website outside their publication, because I think it's reasonable to start thinking about working alongside the traditional publication. Then we'll let the world figure out what really is more useful to them. And in terms of indexing in PubMed, we would, ultimately like to have a ClearScience project, have a DOI, and probably whatever way we represent the actual final quote publication, I don't even like using that word, have a DOI itself.
That is a very good answer. I think Mark R is asking, and Marianne says, thank you. I agree. I think that's a great model. I mean, generally speaking, if you want to really change publishing, you can't all of a sudden turn around and say, no, 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 no. All of you need to stop publishing the way that you've been publishing for 600 years. It's just not going to work that well. But I think your approach is very nice here. Mark is asking about details about pricing. I believe that Sage is all non-for-profit, but I don't know. Can you guys give any details about pricing?
Yeah. So I mean, I think there are a few layers of answering that question. The first is that Synapse is there is no fee to use Synapse, to store data there, to store code there. As we head forward and integrate more with cloud computing, there will be further costs that will be incurred by Sage, by Synapse, and possibly may getting passed on to the user. So if you would want to spin up a cloud compute machine, we would give you the ability to do that. You would have to pay for it, but it wouldn't be sort of paying for the services and apps. It would your compute resources, and on top of that, I think another portion of that pricing, which is a little bit more towards the scholarly community, that we're going to have to think about is longevity of these artifacts. So it's very easy, quote unquote easy, to archive a publication. It's sort of a fixed amount of data, a fixed amount of storage, but once you start talking about large genomic data sets and compute resources, the questions get a little tougher to answer. So that's something that we're most certainly investigating.
That's great. I think Mark is going to thank you. Sorry for putting words on your keyboard, Mark. I'm trying to enable everybody's microphone rights right now. So you should be able to turn on your microphone as soon as I am to your name. But in any case, so there were a couple of harder questions that I alluded to. The couple of harder questions are, what are the types of data that Sage handles? And the second question, which is highly related to, is once you have a data set, how do you know that it's a different or same data set as someone else using a different R package, using a non-Sage system, would actually be able to, it is actually using? So, you know, it's essentially the question, and this is maybe a little bit unfair, but, you know, I don't believe that every scientist in the world knows about Sage, right? And nor does everyone want to use Sage necessarily, or use ClearScience, or use whatever. I believe that there's a plurality of these things. So have you thought deeply about, you know, what it is exactly that you're going to be able to handle or not going to be able to handle in terms of other applications, and the integration and interoperation of those? Because I'm presuming
So
most of your data sets are still coming from Geo.
the great thing about working with software engineers is they always think about generalizability. So the APIs, even though right now we're showing you a technical demo using R. The APIs are generic, right? So in fact, there's a group working for working with Synapse, and it is actually very functional. In fact, some of its functions are actually more functional than our R. There's even a command line. You can actually do it from the bash prompt if you're a Unix propeller head. So we only keep that in mind. In the cases that we showed you, yes, objects from Synapse, but our model is that you ultimately can place any. I mean, you can put a tab delimited text file to a CSV file. In fact, we've been talking a lot about Synapse, whether in fact we're going to encourage people just to put CSV files. Brian sounds like he wants to weigh in too.
Yeah, absolutely. So what we're trying to move towards with a flexible framework, one where users can specify very structured, but really we're leaving it open for people to be able to store whatever types of files, whether that be an R binary file, whether that be a text file, whether that be a piece of code, whether that be a plot, and making all of that accessible again through the Synapse REST APIs. We have a couple of external groups that are working with us to, in fact, share authentication, so their applications can access all of the information that is stored within Synapse within their native environments. So it's a very, very flexible technically. Towards what other types of data lives elsewhere. So a couple of things. Synapse actually doesn't. This is a very confusing, but Synapse actually doesn't store data. Synapse stores information about data, and so a lot of the data that we index is stored elsewhere and is just available through web URLs. When we talk about actually storing data within Synapse, what we're actually doing is storing data in Amazon S3 buckets. And really, to the user who's consuming it, it doesn't matter where it lives. All that's important is that, one, they have the appropriate access restrictions to be able to get at it. So that's another piece of Synapse that we really haven't talked about, but especially dealing with human data, there are some cases where you can't make the information widely available. So you can set access control lists such that you can narrow the scope of who can actually see the data. But as long as you have access to it, no matter where it's getting pulled from, whether it's getting pulled from Amazon S3, whether it's getting pulled from the TCGA web portal, whether it's getting pulled from, or whether it's being pulled from a remote server somewhere.
Yeah, so in theory, if your institution had servers, let's say it's sequencing data that just is frankly too big to push up, quote unquote, Synapse. As long as there's a web link to it, we could basically pointer to it via Synapse. So there's ability built into the model, and that's again a good reason. Well, part of the reason why Brian and I, have enjoyed working here is basically the fact that we have software engineers, in fact, even I working with us.
Okay, so that's great, but the question I was really getting to is, okay, so let's say I have a database like Gemma, and Gemma has is out of UBC. They basically take a whole bunch, if not almost all of the reasonable data sets from Geo, and they do their own analysis, right? Now you guys are going to overlap some of this data because you're also pulling from Geo largely, or not, not completely, but what if, and of course there are the European initiatives. Is in Array Express. Some data is in Geo, and some data is already has been transformed by other projects. Now, I am going to do a search across the web to find all the pieces that I would like to kind of add to, right? So the real question that I have is, okay, oh, wow, what happened here? What am I sharing? Okay, so the real question that I have is, what is the way to really tag these data? How does SAGE envision that there are going to be other environments? Is that you guys doing this whiteboard sharing?
No.
Or is that me?
Looks like someone doing some classification modeling.
Yes, that's not where we want to go today. So you can potentially address that. And Susanna has a question about data collection curation. She's assuming that data sets and analysis workflow ClearScience use comes from Synapse, and this is where we get these submitted and harmonized. Otherwise, all these great functionality won't be possible. So do you see Suzanne's question?
Yeah, we do.
Okay. So, again, my question is, yes, your system, it works possibly better than any other system that I've ever seen in terms of keeping track of the provenance of how the data, work while they're in your system. But if there is data that's analyzed outside of your system, have you thought about basically how to interface with other systems?
Yeah.
Well, I'll let Brian go first.
Yeah, so again, getting back to a little bit about the unique web URL. So with Array Express, with Geo, with TCGA, just as those are three good examples, we don't want to store data. Do is point back to them because that's really where sort of the provenance of what anybody's work should go back. It shouldn't go back to something stored again in Synapse. It should start from an Array Express entry, or it should start from the TCGA. But what we're trying to do is provide sort of a common way for people to access those data and to be able to describe what they've done once they've started working with that data. Transitioning into Susanna's question, which is a really important one, what we're really hoping to do is provide people the tools. So we've tried sort of the en masse curation and normalization route that a number of other groups have gone down, standardizing everything. The issue is most analysts are very opinionated about how their data is processed. And there are a lot of other people in this space already. So what we're really trying to provide is a way for people to access whatever data that they feel like they need and to be able to do custom analyses as they see fit. Sure. So the provenance that we're putting in place certainly would allow that. I mean, obviously that would have to be something that Geo would want to do, but really we could take already we can. The problem is provenance is so young, we don't yet have much built into that. But over time, you'll be able to take an object and say, what are all of the things that have used this object? And so you could do exactly that. You could essentially come up with a list of analyses that have spawned from this that utilize Synapse tools, realizing that there are probably other groups who won't use it.
And as I mentioned, Synapse provides basically pointers, right? Brian specifically made the distinction. Synapse itself is not, when you're accessing a Synapse entity, you're not actually accessing the data. You're actually accessing a pointer to the data. So that pointer generally is an S3 bucket in Amazon, but it could be, because Geo has unique URIs for all their data. It could be the actual piece of data in Geo. And the other thing you can do, and we haven't built this in, well, actually it is built into a large degree, is that, so then the question provenance wise is, let's say that piece of data in Geo, are you sure that's going to be the same, even if the web URI is the same, are you sure that piece of data is going to be the same? Maybe they realized that there was a normalization problem and they put up a new version of those data. Um, you can actually match those using hashes, right? So in fact, that was a thing that I was starting to explore before leaving Duke. Was you can actually digitally fingerprint every piece of data that you use, and in fact, part of the metadata in Synapse is a hash. So that's a unique digital fingerprint for that piece of data that's algorithmically generated from that piece of data. So it's always tied to that piece of data. And you can use that to verify your provenance. So there's always some sort of quote unquote paper trail, in this case, a digital paper trail that you can always refer to. Yeah, I mean,
Very, very important to have data. And these are the numbers you have, but something generated those numbers. Exactly. Mm-hmm. Yep, agreed. That's great. So I only have one other question. So R can be used for many, many different things, and I don't know if it's up to the thousands, but certainly at least hundreds of people have added pieces to R, their own analysis algorithms and what have you. So let's say I have an MRI data set or or a microscopic imaging data set, or something that is very, very different from, a slew of numbers. I mean, it's at its core, I think it is still a slew of numbers, but they certainly mean different things. They are floating points and they are these various things. So let's say I have a data set that doesn't look like microarray data. Would I be able to still use SAGE, or do you envision that ever being part of this? Mhm. Mhm. Okay. Well, we're kind of slowly wrapping up the time here, and unless Lisa has a question. Um, so I want to, from the top, kind of thank both you, Eric and Brian, very much for taking us through this, and I'd like to thank all of the people who participated. Thanks for the questions, and I'm really looking forward to more of these kinds of tools being available in my next set of publications that I have to either write or review. Um, so, I mean, this. Okay, so Lisa actually did have a question that wasn't a thing. So she's asking if whom is the community currently, and how do others learn about ClearScience? All right. I think that was a very, very reasonable answer. Thank you again. And I think, yes. And sharing science is what Lisa is saying. So that is absolutely true.